My name is Bernie Siegel. I'm the Executive Director of the Regenerative Medicine Foundation. We're doing a series of broadcasts with leading researchers and novel research that's going on, things of importance in the field, and doing a general catch-up with some of the advocates, investigators, policymakers in the field of regenerative medicine. I'm very, very pleased today to have with us Dr. Dana Cairns and Dr. David Kaplan of Tufts University that have done some really significant research that was very well reported by Sharon Begley in STAT. And the title of the research is Using Human Brain Tissue in Lab Dishes. Researchers show herpes linked to Alzheimer's. This is significant in many, many ways, and the fact that we have these prominent researchers available to discuss their multidisciplinary approach to this research, where sometimes you start to get from A to B, and then you find yourself at C, D, E, and F. That's the nature of this study that's making some waves, certainly in the circles of Alzheimer's and the impact of pathogens on diseases of the brain. So let's begin. So Dr. Cairns, Dana, please tell me a little bit about this scientific experiment. What's the background of it?
Thank you for having us. This is really lovely and a nice opportunity. So basically, we had this stem cell that we derived in our laboratory, and we had used it for a variety of other studies and applications, and even including some previous Zika research. So an article came out that a population study out of Taiwan that looked at was able to correlate risk, herpes simplex 1 risk associated with Alzheimer's incidence in populations. And so we had initially, it just started out as a fluke, seeing if we could infect ourselves with this virus, and just to see what would happen, because as you probably know, not all cells are easily infectable. You don't always see different kinds of phenotypes. So when we did some of these preliminary experiments, we were just really, I guess, kind of shocked to see what happened. So when you infect them at a very low level, they form these big conglomerate structures that when you immunostain them, or you look at the proteins that are expressed in those big clumps, what you find is that they're full of amyloid, which is the protein expressed in the plaques involved in Alzheimer's. So it was just, I said the word fluke, but in many ways it was like I don't want to speak on behalf of David, but I think, we're not Alzheimer's experts, or at least weren't, not that we are now, but we certainly weren't at the beginning of this study. And so when we started to have these findings, I actually reached out to Dr. Ruth Itzhaki out of Manchester to see if she had any kind of feedback, and she was really wonderful and provided a lot of guidance, and we just sort of went from there and followed the data, as opposed to forming a lot of hypotheses and assumptions. We just sort of went with it to see how it panned out.
That's rather amazing, and it seemed to me that this research study was building upon work, Dr. Kaplan, David, of yours related to biomaterials. And a burning question that I had when I reviewed the paper is the fact that you weren't using what is commonly known as mini brains, but you were using something different, a system that apparently you created using caterpillar silk. And how original is that, and what is the impact of using it? Why this type of biomaterial?
So the biomaterials that we use in our brain model are really critical to provide the correct chemistry, morphology, stability in culture, cell-friendly features, and so on. So the engineering that goes into that is very important, because you need to create two different niches for the cells. You need a region where the neurons and other cells will sort of be able to sit and function well, but also you need a region where these cells can send out axons, make connections and create function. And so we had to build a bioengineered model for that that matched all the requirements, and that covers optical issues. It covers making sure you could put the right ECM component, extracellular matrix components to match the cell needs. You have to have something that will be stable for long periods of time to study, generally to study neurodegenerative diseases. And you need systems that are mechanically matched well enough to the cells to allow them to function the way you want. So there are a lot of design inputs that go into this.
Do you see this as something that will be used by other researchers moving forward as and perhaps a better way of going forward with this type of research?
So I would say it's a different way. I think every system that people are using offers specific insights, and they're all good for certain things. For our goals, though, we needed systems that could be directly imaged, so non-invasive imaging. We needed systems that would be stable in vitro in culture for months and years. And we needed systems that sort of, allowed the biology to work well. And so that's what we've achieved. So a good comparison is, we build our systems from the bottom up. We add the right cells. We know how many cells are there. We put them where we want. So it's a compartmentalized model, so you can track what's going on. And we've already published before even this paper that we can grow these for 2 years without significant necrosis in the model. So this is a huge plus if you want to do long-term neurodegenerative studies. What changed? So we published, for example, a 2-year study on Alzheimer's already with IPSCs and other cells. And that was great. But now with Dana's work, all of a sudden we can shorten that time frame tremendously to screen cells and conditions that we couldn't do before. So this is a major plus in terms of looking at infections related to accelerating the phenotype and understanding eventually how to correlate that to genotype as well. So,
David, thank you for that. So Dana, you didn't use IPSCs, and some of the listeners to this broadcast are very familiar with induced pluripotent stem cells. Was there a reason that you didn't use those cells? You used human-induced neural cells. Explain the difference and why you used the cells that you did.
Sure. So they are somewhat similar. They're both stem cells. One thing with pluripotent stem cells, which I'm sure your audience is aware, is that they can become anything, right? So they're similar to embryonic stem cells. They can become any tissue in the body. With neural stem cells, they're sort of a little bit pushed toward that neural lineage farther. So you're held stagnant in a neural stem fate, so that it's just a little faster. So you don't, and the other benefit is that there is no chance that these cells will differentiate into anything outside of neurons and glia. So in that sense, it's sort of like an expedited differentiation and expedited brains.
You seem to have made a major advancement. It has the neuroscientists excited. It has the Alzheimer's community excited. Where do you go from here to build on this study?
I think it would be really interesting to explore a few more drug opportunities. So as I mentioned, the Valtrex would be ideal for someone at the early stage. So in our system, it really helped to prevent amyloids or to prevent them from getting worse. So I think what would be really interesting would be to use this model to test drugs for those patients, to mimic those patients that are in severe progressive disease. So something that would work for breaking down those plaques as opposed to preventing the formation. I think that would be really a nice model system for that, especially because in many ways it's quite high throughput. You can generate 100 of these donuts in one experiment and test a huge variety of drugs in 3D. So it really lends itself to a wide variety of different types of tests we could do moving forward.
David, it sounds like this could be a blueprint for some future research. What do you make of it? How are you funded to be able to progress this research and take it to the next level?
I think there's a couple of issues. One is we aren't funded for it, so we're writing grants to build on these early discoveries. And I think Dana's spot on. I think we need to expand everything from the patient-derived sources of cells we can develop and enter into the system to figure out how to correlate the responses in the model, genetic sort of predispositions to the outcomes when we screen these various antiviral and perhaps anti-inflammatory drugs. I think that would be ideal, to build a kind of a platform system to be able to do that in a very systematic way, because essentially what this does is shortens the time frame to get really good answers, whether a drug works, literally in a week or two, right? So this allows you to scale it, develop a high throughput or moderate throughput system to really do that for first, like a broader range of questions, but eventually for patient-specific cells that could be developed and put into the system. So I think there's huge potential. Obviously, we have to walk before we run, but that's where we are.
It seems to me you ran before you walked with this research and really made some useful discoveries through innovation and a multidisciplinary approach. I'm wondering if you're dealing with any other collaborations outside of Tufts University itself.
Sure.
Who are you looking to collaborate with, if anyone?
Well, so there's two answers. One, on the brain-like tissue model that we're talking about, we've been collaborating with many labs over a number of years on different questions. So for example, with colleagues at Mass General Hospital, we work on traumatic brain injury with our model to get insight into mechanisms and potential treatments. With colleagues at UPenn at the medical school, we work on literally brain transplants. Can you use our brain model and place that in a damaged part of a rodent brain, and will it improve functional outcomes? So we have a number, and there's many other collaborations ongoing. So we do have some labs that have been working with this different kind of model and really achieving very nice successes with it. So it's hopefully now at the point where we'll start to find additional collaborators, particularly in the Alzheimer's field. As Dana said, we're not experts in Alzheimer's, but we're seeing important findings here. And we do have ongoing, I would call it more traditional Alzheimer's studies with colleagues at Tufts Medical Center who are Alzheimer's experts, but not so much on the infection-related side of it, which is this new direction.
Thank you for that, David. Are there any ethical issues that surround this research in using your tissue-engineered system as opposed to human mini brains, where there's concern about what's actually developing and self-assembling in the dish? And bioethicists are weighing in on that and wondering if this is encroaching on some ethical boundary or societal boundary. What's the difference?
Well, from my perspective, for what we're really talking about here, we're developing a platform to rapidly screen drugs to help patients. So in terms of ethics, I feel very comfortable that, for the scope of a particular kind of infection, an Alzheimer's type of response, this gives us a new way to screen medications to potentially help these patients. So I think it would almost be, in my mind, unethical not to pursue that, right? Because of what these folks have to go through. Using the model in other manifestations that I mentioned, each of those has to be vetted very carefully, and make sure we or others are doing things in a way that's both meaningful and helpful. And that's for obviously our colleagues and others to weigh in on. But in terms of what we're talking about here for Alzheimer's-based drug discovery, drug screening, I'm very, very comfortable.
I'm wondering if you're looking to commercialize this technology in any way at this point.
Right now, we just like to see if we can make something that helps people. We do have, overriding patents on our designs and our materials and our systems. I'm not going to get into that today, but that is out there. Because again, we developed the model many years ago, but this is a whole new direction for it. And I think Dana undersold at one point here. I think just take herpes, and that's negating all the other, I'll call it neural infectious agents that are out there, right? We're just talking about one today, and the propensity of infections throughout the human population is overwhelming already, most of whom don't even know they have the infection, right? They're just carriers. So even if people develop screens to know if you're a carrier and do that very proactively, would be a big help, because then you would start to say, okay, I'm the carrier, I don't know, but then maybe I do need to start looking at, what kinds of drugs I need to look at to reduce the neuroinflammation, reduce the, the virus that may be carried but sequestered and not active. And I'm hoping this will spur new discoveries. How do you find new drugs to get into nerves to attack these viruses? That's not trivial either. So to me, it just raises some really important scientific questions that have direct potential impact on a large segment of the population today.
If other research, yes, go ahead, Dana.
Just to cut, because David made a point that I think is worth mentioning. So he mentioned that, like you said, we looked at one pathogen, right? So we just looked at herpes, and it was able to cause this type of phenotype. But there are other emerging studies that suggest other pathogens. So there's, I'm not sure if you're familiar with Porphyromonas gingivalis. It's a type of bacteria found in your gums. So it's like causes gingivitis essentially. That's another pathogen that's been recently, there's this idea that is connected to Alzheimer's as well. It can, in theory, travel up through kind of the nasal canals and into your brain and cause, AD-like symptoms. So I think another cool collaboration would be to use this system to infect with other pathogens that have a presumed role in this process. There's other strains of herpes that have been suggested as well. HHV6 is one that's being considered. So in terms of collaborations, I think other groups that particularly microbiology groups that are particularly interested in how their pathogen of choice might be implicated in this disease pathogenesis, that would be, groups we'd be interested in working with as well.
If I'm correct on this, that the study of pathogens in general as a direct cause of Alzheimer's is fairly recent. And the funding from the federal government for this is also fairly recent. This is you're breaking new ground with this. Is that correct?
I think we're building upon other people's, like the idea that it's possible has kind of been in the groundwork for some time, but I think in terms of making the direct connection, we're kind of in the midst of it. So it's a really exciting time to be a part of science in this field specifically.
Dana, I think you've understated what is being described about your particular research. This seems to be the first research to ever show a direct connection between the herpes virus and Alzheimer's. Is that correct?
I would be remiss if I didn't credit the people before us who have done such great work. So I think it's really exciting that it's all kind of tied together with previous studies and things people have been thinking about for a while. But
off the record.
But let me add two things on that. I think there's there's some prior work out there that strongly made these links already. I think what we've been able to do here is we had some new tools, the cells Dana generated and the 3D tissue engineer model that allows it to be what I would call a more practical, directly relevant connection that should accelerate the rate of hopefully interest and discovery from here. And I would add one more caution here. We're all living through a pandemic right now, and there's very clear evidence that not only the lungs and the intestine, but the human brain is becoming a receptor for these viruses as well, the COVID. And so this may just be the beginning of another kind of infectious agent that may have downstream long-term consequences towards neurodegenerative something, right? Whether it's Alzheimer's or other, we don't know. But we think it's an important topic to bring to the fore now, that we should start to look at this kind of infectious agent as well and determine ahead of the game how to control it in the brain as well to hopefully avoid long-term problems in the population.
That's a very profound remark, David, because I have a family member that has recovered from COVID, but went through a bout of dementia, severe dementia, was intubated, was on a respirator, and it seemed like an inflammation of the brain. It was really something shocking and something that has been very hard in her convalescence. So as COVID presents so many unknowns to us, the thought that a pathogen might have impact on multiple organs is very significant. So I appreciate you making the connection, because I was afraid to even venture into the speculation, but it occurred to me.
Look, I think it raises, my perspective is, if nothing else, it raises awareness in the broader population while they're all sensitized to viral epidemics going on, that it's not just the immediacy of these infections that you worry about. It's the long-term implications that we need to really dig into and understand. And I think people have not really given that their due in the past. You get these one-off papers, you get a few labs that have spent some time on this, which has been critical to us in terms of leaning on some people that we really value their inputs on here, like Ruth. But I still don't think the general medical profession really sees that connection. So if nothing else, I'm hoping this spurs two things. One, people to start looking in this direction a lot more vigorously to make those connections, not just to Alzheimer's, but there's, I don't want to get into details, but you can talk about things like Bell's palsy. You can talk about things like just general dementia. You could talk about things like depression. I mean, the list goes on and on, Bernie. So I think it's really important to raise that awareness, number one. Number two, and Dana and I can both speak to this, we are a very open, collaborative lab. We work with many people all around the world, and we would love to continue to build momentum on the model and the approaches that are coming out of the lab with experts in any area if they have the interest. That remains to be seen, but hopefully we can build on this. Obviously, we need to raise resources to do this right, but I think hopefully the momentum will take off from where we are. So that's my perspective at this point.
I think you're both brilliant, first of all, and patient, which I appreciate being patient with this. As another gardener has decided to clip some more plants in the front yard,
Can you send them up here to Boston? My yard is a mess.